Hello everyone. The topic that will be dealt through this video is regulation of oxidative phosphorylation. This is Dr. P. Mary Anupama, Head of the Department of Biochemistry, St. Joseph's College for Women. The learning outcomes of this particular topic would be, by the end of the lesson, the learners, they'll be able to illustrate the need for regulation of oxidative phosphorylation. They can comprehend the impact of cellular energy needs and oxidative phosphorylation. Then they'll be able to narrate the impact of ischemia, which is lack of oxygen, and the IF1 protein impact in ischemia. They'll be able to explain the underlying reason for uncoupling of oxidative phosphorylation to produce heat in the case of infants and hibernating animals. Then they'll be able to discuss the combined effect of ATP producing pathways on oxidative phosphorylation. To attain those objectives, the contents that will be delivered in this lecture would be introduction, followed by regulation by cellular energy needs, regulation by inhibitory proteins as in ischemia, inhibition by uncoupling for heat generation, and then regulation by ATP producing pathways. Introduction. In aerobic cells, we see that oxidative phosphorylation becomes the main source for production of our energy need, that is ATP. A glucose molecule, if it is completely oxidized into carbon dioxide in aerobic cells, then it could result in the production of around 30 to 32 ATP. So let us see how we are getting the yield when glucose is completely oxidized. So here is the table. You have the process over here, the product which is formed, and finally the ATP that you get from this product. When a glucose is oxidized, it enters glycolysis. It gets converted to pyruvate, which is oxidized, then acetyl-CoA, which is oxidized in the citric acid cycle. These products which are formed, they enter the electron transport chain, and finally you get ATP synthesis. So first is glycolysis. Glucose enters glycolysis, where you get 2 NADH and 2 ATP. From these NADH, you get either 3 to 5 ATP, and this ATP directly we have put up here. Next is pyruvate oxidation. One glucose molecule will yield 2 pyruvate molecules. So if 2 pyruvate molecules are oxidized, you will get 2 NADH, and this is taking place in the mitochondrial matrix. Glycolysis will be taking place in the cytosol, and pyruvate oxidation is taking place in the mitochondrial matrix. So from 2 NADH you get 5 ATP. Next is acetyl-CoA. So acetyl-CoA oxidation is taking place in the citric acid cycle. So from 2 pyruvate which are produced from glucose, we have to see that it is 2 acetyl-CoA which are produced per molecule of glucose. So as these are getting oxidized, you see that it is 6 NADH which are produced, and this is also taking place in the mitochondrial matrix. So from 6 NADH, you get around 15 ATP molecules. Apart from this, you also get in the citric acid cycle 2 FADH2, which will result in 3 ATP molecule formation, and 2 ATP molecules or 2 GTP molecules that will result in 2 ATP. If you sum up all these things, the net ATP yield per glucose molecule would be 30 or 32. This is in aerobic cells. But when you look at the anaerobic environment, as you see in the lactate fermentation, the amount of ATP produced per glucose molecule after it enters glycolysis, it will be very less. It is just 2 ATP molecules. The other variation is you see that when a coenzyme A derived palmitate, if it is getting completely oxidized, the amount of ATP that is produced per molecule of this one would be 108 ATP. So you see that there is a vast variation with respect to ATP that is produced during catabolism under aerobic oxidation pathways. So all these will be leading to electron transfer to oxygen and oxidative phosphorylation. You see that this particular thing, that is electron transfer to oxygen and then ATP production, that is very important and it is highly regulated. This happens based upon the cell needs. That means when a cell is very active, that will need lot of ATP, while a cell which is not so actively involved, that will be requiring less of ATP. So this is how it will be varying. This is how based upon the need of the cell, there will be a fluctuation in ATP production, which is nothing but the regulation which we will be studying. First regulation is oxidative phosphorylation, which is regulated by the cellular energy needs. So you see that the rate of oxygen consumption which happens in the mitochondria, it is directly related to the availability of ADP. This ADP availability, it is regulating the rate of oxygen consumption directly. And we know that ADP is nothing but the substrate for phosphorylation. That means ADP upon phosphorylation will result in the formation of ATP. Therefore, the rate at which the oxygen consumption is taking place, that directly tells us how much of ADP is available. Okay, the rate of oxygen consumption will tell us how much ADP is available for phosphorylation. So this is the acceptor control of respiration. And there are few animal tissues wherein the ratio of maximal rate of ATP induced oxygen consumption is seen, while there are few where is total absence of ADP. And if you take the ratio, it is around 10. Maximal to absence is a ratio of 10. Apart from this oxygen consumption, you also have another measure wherein you see the energy status of a cell. This is done and you call it as mass action ratio. We call it as mass action ratio. And it is expressed in terms of ATP ADP system, wherein you see the concentration of ATP, you calculate the concentration of ATP versus availability of ADP as well as inorganic phosphate. If you have more of ADP, obviously the rate of respiration will increase, leading to increased synthesis of ATP. And this rate, it will be fluctuating based upon the energy needs of a particular tissue, which means that ATP synthesis will be very high in tissues which require lots of energy. High energy demanding cells, they'll require it. That means the cells are highly active. Lots of activities are taking place within the cell, and to meet the energy needs, there is a high rate of ATP synthesis. Next type of regulation is inhibitory protein presence, which prevents ATP hydrolysis during ischemia. Yes, it is preventing ATP hydrolysis during ischemia. So when a cell is deprived of oxygen, that condition you call it as ischemia. So this is seen during heart attacks or a stroke, and the electron transfer to oxygen, it is affected. And you also see that if electron transfer to oxygen is not taking place, the pumping of protons, it is also affected. And the proton motive force, that is getting affected also. So if the proton motive force which is required for ATP synthesis, if it is affected, then you see that the ATP synthesis which is done by ATP synthase gets affected. Under such deprived conditions, what does this ATP synthase do is it starts operating in a reverse way. It is supposed to synthesize ATP, whereas it starts hydrolyzing the ATP so that it can initiate something to pump proton outwards. And thereby what happens is this will lead to a drop in the ATP concentration within the cell. So here protons are pumped outside, and this will affect the pH within the mitochondria. The pH drops to around 6.5. Under such conditions, you have protein inhibitor IF1 which is stimulated. This IF1, its role is to prevent that hydrolysis of ATP by ATP synthase. IF1 is full form as it is protein inhibitor IF1, and it is 84 amino acid peptide which initially exists as a monomer. At this particular pH, when the pH drops down to 6.5, it starts dimerizing and you get a dimer of IF1 at pH less than 6.5. And this is seen in cells that are deprived of oxygen. So when the cells are starved, what will happen? As I have told you, this ATP, it starts getting hydrolyzed. And the cells are starved because of the oxygen, deprived in oxygen. The cells will be starved of energy from glycolysis because ATP hydrolysis will be taking place. And parallelly you see that there is production of pyruvic acid or lactic acid that is taking place. Mainly this pyruvic acid and lactic acid, they will be lowering the pH in the cytosol of the cell as well as in the mitochondrial matrix. So this drop down because of the pyruvate as well as the lactate accumulation, that is causing the dimerization of this IF1. So what does this IF1 do is it again starts binding to two molecules of this ATP synthesis. It starts binding to the molecules of ATP synthesis. So once this bind to it, then the ATP synthesis will stop hydrolyzing ATP. So thereby it is preventing wasteful hydrolysis of ATP. Now this will last for a while. And meanwhile, when the pH of the cell is restored, that is, it starts gaining pH above 6.5, then this will come off. So what's happening? Again, ATP synthase is free and it will start restoring its function, which is nothing but synthesis of ATP. So this will start functioning or it will start synthesizing ATP. So this is how IF1 binds to ATP synthase and prevents hydrolysis of ATP under ischemic conditions. Next, very interesting role or a very interesting regulation is heat generation by uncoupled mitochondria. This will be finding in the adipose tissues, the brown fat which you find in the newborn mammals. Okay, the newborn mammals, they have this brown fat as the adipose tissue. And oxidation of this brown fat, this will lead to heat production. Yes, it will lead to heat production and it does not lead to ATP synthesis. So this adipose tissue, it is specialized that it has lots of mitochondria in it and also it has cytochromes which have heme groups. Those heme groups, they are involved in absorption of visible light very strongly. So these mitochondria of the brown adipose tissue of the newborns, you see that they are just like our normal mitochondria only. But the thing is, they have one specialized protein in their inner membrane, which you call it as the thermogenin or the uncoupling protein. It is thermogenin or the uncoupling protein. So what is the role of this particular protein is, this will start providing a separate channel for the protons in the inner intermembrane space to go into the matrix. So instead of the protons being pumped through the FOFN complex through which ATP synthesis will be taking place, the FOFN complex is nothing but the ATP synthase. So the protons have to be pumped through that. Instead, what will happen? The protons are pumped through this particular channel. It will start acting as a channel, thermogenin. And the protons, they enter into the matrix. Under such situations, you see that the energy of oxidation, whatever that is there, it is dissipated as heat. And that is used by the infants to maintain their body temperature. The body heat of the infants is because of the brown adipose tissue. And the next one is, you also find them same phenomena happening in the hibernating animals also. So there also it is heat production which is required. Here is a diagrammatic representation of heat generation which is done by uncoupling mitochondria. Here you see, as the electrons are passed through the electron transport chain, the reducing equivalents, that is the protons, mainly the protons, they are pumped into the intermembrane space. Here they will be accumulating. Now these protons are to be sent back to the matrix, and this happens only through the ATP synthesis. When if it is transferred like this, you will be getting ATP synthesis taking place. Rather, in newborns, these mitochondria have thermogenin, which is the uncoupler. Once the uncoupler binds here, the action of this ATP synthase is stopped. Rather, it becomes the channel diverting the protons to enter into the matrix. During this process, as the protons are being pumped back through this, heat is generated. So this heat generation is because of uncoupling of mitochondria. The last and important regulation is regulation at the ATP producing pathways, which coordinately regulate the oxidative phosphorylation. So where do you get ATP from? We'll be getting it from the biological molecules. As the biomolecules are, or the precursors, these precursors, as they are being catabolized through various pathways which work together, ATP is produced. So here you have the rate of electron transfer, oxidative phosphorylation, citric acid cycle, oxidation of pyruvate, glycolysis, all these are regulated by the relative concentrations of mainly ATP as well as ADP. So the concentration of ATP and ADP is regulating all these events. The rate of electron transfer and oxidative phosphorylation, both these things, their rate, it is increased with increase in ATP consumption. You know that the end product of oxidative phosphorylation is ADP. If the end product is consumed, so obviously these previous reactions they'll be continuously operating. So here the rate at which pyruvate is oxidized through the citric acid cycle, this will also increase because ATP is consumed. So obviously from glucose to pyruvate, pyruvate to citric acid cycle, this speed also increases. And this in turn will lead to increase in electron flow into the respiratory chain. And thereby here you can see that the rate of pyruvate formation is also increased. If pyruvate is more, the citric acid cycle operation will be more. Thereby the electron transport chain will be operating little faster, more electron flow, and then again more ATP synthesis. If ATP is consumed, then only all these things will increase. So this we call it as the acceptor control. We have already seen this acceptor. This acceptor control, this is slowing down the overall process. We have seen that acceptor control is nothing but the levels of ADP. Levels of ADP, because ADP is the acceptor for inorganic phosphate, and finally it is resulting in the formation of ATP. So if ADP concentration is less, ATP also will be low. So the overall synthesis of ATP will decrease. And apart from this, ATP is also acting like an allosteric inhibitor. So it's an allosteric inhibitor of glycolytic enzymes like our phosphofructokinase 1 and even pyruvate dehydrogenase. So both these enzymes, they are allosterically inhibited by this thing. That means if concentration is high, these two enzymes will be affected. So obviously glycolysis as well as citric acid cycle, they will be slowed down. Apart from ATP, even the citrate is affecting phosphofructokinase. This is also inhibited by citrate. And citrate, we know that it is the first intermediate of citric acid cycle. So when the cycle is operating, obviously citrate will start, its production will increase and citrate will start accumulating within the mitochondria. And apart from that, as it is accumulating in the mitochondria, it will also start spilling into the cytosol. So its level in the cytosol will also increase, thereby affecting glycolysis. So when concentration of ATP as well as citrate increases, okay, they can cause obviously allosteric inhibition of phosphofructokinase. So this is very much more than the sum of individual effect of both the enzymes. That means if we individually sum up the extent of inhibition caused by ATP and individually caused by citrate, and then if you add both the things, that is ATP plus citrate, and if you try to inhibit this one, you see that it is much more than the individual impact. So that is how both the ATP as well as citrate, they are affecting phosphofructokinase 1. If they are more, if the end products are more, obviously this particular enzyme will stop, which in turn will slow down ATP synthesis or oxidative phosphorylation. The references that we have used in the preparation of this content include Leninger's Principles of Biochemistry Biochemistry by U. Satyanarayana. Then Harper's Illustrated Biochemistry. All these are used. To summarize what we have learned, first we have seen net ATP yield from glucose, which would be 30 to 32. And then we have seen that under anaerobic conditions, the ATP yield is reduced. It's just 2. And we have seen that if it is palmitoyl-CoA, it goes up to 108 ATP yield. And we have seen the various regulatory mechanisms. Oxidative phosphorylation, it is regulated by the basic cellular needs. Okay, here you see that availability of ADP is important. If ADP is available, oxidative phosphorylation will be taking place, leading to ATP synthesis. Other regulation is by the inhibitory protein IF1, which prevents ATP hydrolysis during ischemia. In case of ischemia, we have seen that ATP synthase starts hydrolyzing ATP. Therefore, its regulation is important and is done by IF1. Then in newborns and hibernating animals, we have seen that it is uncoupling that is done. That means here oxidative phosphorylation ATP synthesis will not be taking place. Rather, electron transport chain happens, proton gradient is produced, but that proton motive force is used for generation of heat. And lastly, we have understood that yes, it is a networking that is happening. Many factors keep influencing the synthesis of ATP, mainly the ATP concentration itself, which is produced from various pathways. Thank you for joining. Please study the material and then prepare for the assessment. Thank you.